Chandipura virus, CHPV, is a highly pathogenic virus belonging to the Rhabdovirus family and the genus Vesiculovirus. It is recognized as an important cause of acute encephalitis syndrome in children because of its extremely rapid disease progression and high mortality. From a clinical microbiology perspective, understanding its biology, transmission, diagnosis, and management is essential, since the infection represents a pediatric medical emergency, with neurological deterioration occurring within a very short period. The epidemiological profile of CHPV shows that it predominantly affects children younger than 15 years, with the most severe disease commonly seen in those between 2 and 5 years of age. First identified in Nagpur, India in 1965, subsequent outbreaks in Gujarat and Andhra Pradesh report in case fatality rates ranging from 55% to 75%. The disease progresses rapidly, with many deaths occurring within 24 to 48 hours after the onset of symptoms, leaving only a narrow window for clinical intervention. CHPV is transmitted through the bite of infected sandflies, particularly Phlebotomus papatasi and Sargentomia species. Vertebrate animals such as pigs, rodents, and cattle serve as environmental reservoirs that help maintain the virus in nature. Importantly, there is currently no evidence of human-to-human transmission, making vector control and environmental surveillance the primary preventive strategies. The virus possesses a characteristic bullet-shaped morphology measuring approximately 150 to 165 nanometers in length. It contains a host-derived lipid envelope studded with glycoprotein spikes, while the interior consists of a helical nucleocapsid enclosing a negative-sense single-stranded RNA genome of about 11 kilobases. This genome encodes 5 essential viral proteins: nucleoprotein N, phosphoprotein P, matrix protein M, glycoprotein G, and the large polymerase protein L, each playing a critical role in viral replication, assembly, receptor binding, and transcription. CHPV demonstrates unique multiplicity of infection, MOI-dependent pathogenicity. At high MOIs, viral replication is rapidly followed by extensive cytopathic effects that destroy infected host cells. This explosive cell death limits further viral replication by eliminating the very cells required for viral production. Such rapid host cell destruction contributes significantly to the aggressive clinical course observed during infection. Within infected cells, the viral N and P proteins undergo liquid-liquid phase separation to generate cytoplasmic inclusion bodies, IBs. These membrane-free structures function as specialized viral replication factories where genome replication and transcription occur efficiently. Inclusion bodies also provide a protected microenvironment that shields viral replication from several host antiviral defense mechanisms, allowing uninterrupted viral multiplication. CHPV further suppresses host immunity by actively recruiting important antiviral proteins such as PKR and TIA1 into its inclusion bodies. By sequestering these molecules, the virus prevents the formation of normal stress granules and avoids translational arrest. Consequently, viral protein synthesis continues efficiently while the host's innate antiviral signaling pathways remain functionally impaired. The clinical course begins after a short incubation period of approximately 0 to 2 days, followed by sudden onset of high-grade fever and repeated vomiting. Neurological manifestations rapidly develop, including seizures, altered consciousness, and encephalopathy. Without immediate medical intervention, progressive cerebral edema and brain herniation may develop, frequently leading to death within 48 hours of symptom onset. Laboratory diagnosis depends greatly on the timing of specimen collection. During the early viremic phase, usually within the first 4 days of illness, real-time RT-PCR performed on serum or cerebrospinal fluid (CSF) remains the diagnostic gold standard. Because of the extremely rapid disease progression, treatment decisions often need to be initiated before laboratory confirmation. IgM ELISA becomes useful after the first week for retrospective diagnosis and epidemiological surveillance. Currently, there are no approved antiviral drugs specifically targeting CHPV. Therefore, patient survival relies entirely on early aggressive supportive management. Clinical priorities include reducing raised intracranial pressure with Mannitol, controlling seizures using anti-epileptic medications, maintaining adequate airway, breathing, and circulation, and ensuring strict hemodynamic stability during the critical first 24 hours of illness.